Extraction method of crude drugs. BSc third year. Extraction method of crude drugs. Extraction is defined as the process of isolation of soluble material from an insoluble residue, which may be liquid or solid, by treatment with a solvent on the basis of the physical nature of crude drug to be extracted. That means liquid or solid. The extraction process may be liquid or solid extraction. The process of extraction is controlled by mass transfer. Mass transfer is a unit operation which involves the transfer of mass of soluble material from a solid to a fluid. If a crude drug particle is immersed in a solvent to be used for extraction, the particle is first surrounded by a boundary layer of the solute. The solvent starts penetrating inside the particle and subsequently forms soluble of the constituents within the cells. Escapes of this dissolved constituent through the cell wall and through the boundary layer takes place. The process is continued till equilibrium is set up between the solution in the cells and the free solution. Few important factors which affect the mass transfer are agitation and temperature that increase the concentration gradient to bring about the efficient extraction. Size reduction of the crude drug increases the area over which diffusion can occur. Overall extraction is also dependent on the selection of the method of the extraction and the solvent selected for extraction. Extraction method of the crude drugs. In the third point, the majority of the small scale extraction process of maceration and percolations are generally slow and time consuming and also give the inefficient extraction of the crude drugs. Maceration process involves the separation of medicinally active portion of the crude drugs. It is based on the immersion of the crude drugs in the bulk of the solvent or mixture. Solid drug material is taken in storage container as shown in the figure below. These are the general techniques and mostly applied for galenical preparations. The sole purpose of such basic extraction procedures for crude drugs are to be obtained from the therapeutically described portion and eliminate the inert fat material of the treatment with a selective solvent known as menstruum. Such type of extraction procedures also play a decisive role for the qualitative and quantitative composition of the extract. The standardized extract thus obtained are further proceed for inclusion in other dosages from such as tablets and capsules containing several groups of the plant's metabolites. These extracts are also utilized for isolation and characterization of therapeutically active chemical constituents used in modern medicines. General procedure. Plant material placed in closed vessel, then all of the selected solvent added, solid residue pressed strained and expressed liquid mixed, then clarified by subsidence or filtration. Magnetization method is modified to multiple stage extraction to increase the yield of the active ingredient in the extract. The crude drug material is charged and in the extractor, which is connected with the circulatory pump and spray distributor along with the number of the connected tanks to receive the extraction solution. Percolation, as the term indicates, the percolation is continuous flow of the solvent through the bed of this or crude drugs material to get the extract. In this process, first the powder drug is treated with sufficient moisture to make it uniformly wet. Damp material is allowed to stand for about 15 minutes and the transfer to the precalator, which is generally a V-shaped vessel or often at both ends. To each sufficient moisture is added to saturate the drug. The lid is placed on the top. When the liquid starts dripping out from the outlet of the percolator, the lower opening is closed. Here is the diagram that shows the method. Modified percolation. The conventional percolation process is modified, especially when the solvent is dilute alcohol. In case when the strength of the alcohol needs to be unaffected by concentration of the extract, percolation is continued and the first quantity of the percolate is collected and set aside. The subsequent quantities of the percolates are collected, concentrated, and lastly, the first volume of the percolate is added in the final product. Surface extraction. Surface extraction is the process of continuous extraction in which the same solvent can be circulated through the extractor for several times. This process involves extraction followed by evaporation of the solvent. The vapors of the solvent are taken to be a condenser and the condensed liquid is returned to the drug for continuous extraction. Here is the diagram that shows the continuous extraction. Continuous extraction. Surface affairs are designed for such continuous extraction consists of a body of the extractor attached with the side tube and the siphon tube as shown in the figure above. The extractor from the lower side can be attached to the distillation flux and the mouth of the extractor is fixed to the condenser by standard joints. Supercritical fluid extraction. The supercritical fluid extraction is a comparatively recent method of extraction of crude drugs. Certain gases behave like a free-flowing liquids or supercritical fluids. At the critical point of the temperature and pressure, such supercritical fluids have a very high penetration powers and extraction efficiency. This principle was first used in the food packing industries for the deodorization of the packed food products. The gases like carbon dioxide are held as a supercritical fluid at the critical point of 73.83 bar pressure and 31.06 degree centigrade temperature. At this critical point, carbon dioxide CO2 behaves as a liquefied gas or free-flowing liquid and assists the extraction of the phytochemical constituent from the crude drugs. The phase diagram of carbon dioxide indicates the characteristics areas of the deodorization, extraction, and fractionation. Superficial extraction unit. Type of extraction. Number of different types of methods are used for the extraction of the herbal drugs. And the extract are used for different purposes, ranging from the internal administration, external use for further purification of the phytopharmaceuticals, or for its semi-synthetic conversion to the same therapeutically more active compounds. Aqueous extract. These are the extracts which are medicinal preparations intended to be used immediately after preparation or to be preserved for use. The following methods are generally more in utility for this preparation. Decoction. This is an ancient and more popular process of extraction water-soluble and heat-soluble constituent from the crude rock by boiling in the water for about 15 minutes. The boiled crude rock water mix is then cold filtered and sufficient volume of the cold water is passed through the drug. Infusion is generally a dilute solution of the readily soluble constituent of the crude drug. It is nothing but a type of periodic maturation of the drug with either cold or boiling water. Here is the soft extract and dry extract. This extract, which are produced as semi-solid or liquid of the Friday constituent, are termed as soft extract. These extracts are used in the variety of doses and ranges from the ointment suppositories can be used in the preparation of some other pharmaceutical. Dry extract are also known as the powder extract or dry powders. The total extract obtained by using suitable process of extraction are filtered, concentrated, preferably under vacuum, and dried completely.